Good morning, my dear students. Today you are learning the last cell organelle in the cell under the chapter Cell, the Unit of Life. The name of that last cell organelle is nucleus. So this nucleus is a double membrane bound cell organelle present in almost all the cells. The nucleus is present in almost all the cells and it is a double membrane bound cell organelle. Some exceptional cases, this nucleus is absent like in animal cell, like erythrocytes, RBCs in certain mammals. Certain mammals like the human being, RBCs do not have a nucleus. Whereas in the plant, sieve tube do not contain a nucleus. This is a neat level question. We are ignoring at this time. When question arises in the neat level, at that time we will write the wrong answer. So kindly the student has to be focused certain classes. So let me to tell you once again. The last cell organelle you are learning is nucleus. And nucleus is a double membrane bound cell organelle present in almost all the cells. Exceptional cases like a sieve tube of a plant cell and erythrocytes or RBCs in mammal, certain mammals. Because camel and llama contain nucleus. As I said that neat level, during the neat level class we will discuss in detail about it. Okay. Whereas this nucleus was discovered by Robert Brown. Robert Brown is a scientist who have been discovered this nucleus. And inside this nucleus, a colored thread-like structure will be present, and that is called as a chromatin. That was stained and discovered later on. That is an all new level. We will discuss it in a later periods. So the structure of nucleus consists of this. This is the structure of the nucleus. This nucleus is made up of with the membrane bound cell organelle and two membranes are present. The outer membrane, it is said to be as an outer membrane and this is said to be as an inner membrane. Outer membrane and inner membrane. So outer membrane and inner membrane will be present. In between the outer membrane and inner membrane there is a space and that space is called as a peri-nuclear space. Peri-nuclear space. And this is a round ball-like structure. A ball hole hole that is considered to be as a pore called as a nuclear pores will be present like this. And these nuclear pores are octagonal in shape, N2 sided, 8-sided octagonal pores through which the material is going to be taken into the nucleus and sending the material from nucleus to the cytoplasm. Understand? So, exchanging of material from a cytoplasm to the nucleoplasm and from nucleoplasm to the cytoplasm is taking place through the help of such a type of pores called as nuclear pore. Around 1000 nuclear pores will be present for one nucleus. 1000 nuclear pore for one nucleus through which protein as well as mRNA is going to be taking exchange. Whereas the next one is nucleolus. This nucleolus is round circular structure without a membrane present inside the nucleus. They may be one in number or more than one in number. 2, 3, 4 will be there. Understand? Nucleolus will be present and this is mainly made up of the calcium as well as an RNA as well as a protein. Whereas inside this there is a fluid-like material will be present and that fluid-like material is called as a nucleoplasm. The fluid-like material which is present inside the nucleus is called as nucleoplasm. And this nucleoplasm consists of all the mineral nutrients, proteins, and stored food material will be present within this nucleus itself. Right? That is of nucleoplasm. Then remains is chromatin network. One should understand here. Students will get confused about the chromatin network as well as a chromosome. So chromatin, chroma, color, tina, thread, color, thread. Okay, so chromatin network. It is nothing but coiled DNA. DNA it is. So DNA is made up of a polynucleotide. You know very well that polymerase polynucleotide, and this thread is a DNA. And that we cannot use a word DNA because the DNA is made up of the polynucleotide. So instead of that, they are using the word chromatin, and this chromatin is available during interphase stage of every cell. One should understand that interphase cell division nuclear. When the cell is at rest, that stage is said to be as the interphase stage, and in that interphase stage, the chromosomes present in the cell will become divided and forming into a thread-like structure called as a chromatin. So, in general, we use the word chromatin network. So these chromatin are of two types. One is called as a euchromatin and another one is called as a heterochromatin on the basis of stain and on the basis of their function. What is this stain and function? If the chromatin network is lightly stained, and it is transcriptionally active, that is the function. What is this transcription? You know very well that transcription is nothing but biosynthesis of mRNA from DNA. You will be knowing that DNA containing particular information on it to synthesize a particular protein. ಇವೆಲ್ಲ ನಿಮಗೆ ಅರ್ಥ ಆಗಲಿಲ್ಲ ಏನೇನು ಹೇಳಕತ್ತಾನ ಸರ್ ಅಂತ ಇವಾಗ ಒಂದು ಎಕ್ಸಾಂಪಲ್ ಕೊಡ್ತೀನಿ ನಾನು ನಿಮಗೆ. Here it is. Here is a keratin protein. It is a protein, right? These hairs are growing. Keratin protein. Hair is growing. How it is growing? The cell which must going to be synthesize the hair, the protein. However, the protein will be synthesized, the hair grows. If the protein synthesis less, hair do not grow. ಎಷ್ಟೋ ಜನದ ಪ್ರಾಬ್ಲಮ್ ಇದೆ, ನನ್ನ ಕೂದಲು ಉಚ್ಚಕತ್ತವ, ನನ್ನ ಹೇರ್ ಬೆಳಿವಲ್ಲು, ನನ್ನ ಹೇರ್ ಜಗ್ಗಿ ಬೆಳಗತ್ತವ, ನನ್ನ ದಪ್ಪದವ, ನನ್ನ ಬೆಳಗಾಗತ್ತವ. This is all due to a protein called as keratin protein. Keratin protein ತಿನ್ಬೇಕು ನೀವು. ಅದನ್ನ ತಲೆಗೆ ಹಚ್ಕೊಳ್ಳೋದಲ್ಲ. ಎಷ್ಟು ಜನ ಹೇಳ್ತಾರೆ ಹೇಗೆ ಗೋಳದ ತಲೆಗೆ ಹಚ್ಕೊಳ್ರಿ ಕೂದಲು ಬೆಳಿತಾವ, ಏನು ಬೆಳೆಯಲ್ಲ. You should eat. I will not tell that what is important for growth of the hairs, which containing keratin in it. That is the secondary. So what we want to study is chromatin. It is a keratin. It is also keratin. This is going to be grow ಆಗಕತ್ತಾವ, ಬೆಳಕತ್ತಾವ. ಅಂದ್ರೆ ಏನು? ಅದರ information ಬೇಕು. That information is present on this gene. The gene has to produce the mRNA. The gene which containing information will copy on the mRNA, and that mRNA has to be come out through the help of the nuclear pore where ribosomes are present, and then protein synthesis will take place. This is the phenomenon. So biosynthesis of mRNA from DNA, that is called as transcription. So when transcriptionally active chromatin is called as euchromatin. Nodri istella ಗೊತ್ತಿರಬೇಕು. So once again I am telling you that chromatin which is lightly stained and which is transcriptionally active is called as euchromatin. Whereas a chromatin is darkly stained and it is transcriptionally inactive, that is called as a heterochromatin. That is about chromatin network. So this is the entire history about this nucleus. Let me to tell you once again about the nucleus. Nucleus is a double membrane bound cell organelle, and the nucleus will be present in all the cells. In exceptional cases, the nucleus is absent like a human RBC as well as a tube cell of plant. And nucleus number based cells are identified like a mononucleate or uninucleate, binucleate, trinucleate, that is the secondary. And this nucleus was discovered by a scientist called as a Robert Brown in the year 1831. And its structure consists of like that. The first structure is a membrane, second one is the chromatin, third one is a nucleolus, nucleoplasm, then nuclear core. So among these, outer membrane and inner membrane will be present, and in between that there is a space that is called as peri-nuclear space, that is about the membrane. Inside that there is a ball-like structure, we call it as a nucleus without any membrane. It is not a membrane. It is mainly made up of the calcium, rRNAs, ribosomal RNA, rRNA. It is a protein. That is it, nucleolus. Then next comes nucleoplasm. The fluid-like material present inside the nucleus, that is called as the nucleoplasm, and it contains protein, mineral and nutrients. Then there are octagonal shaped pores will be present about 1000 in a number per nucleus, that are called as a nuclear pore, through which exchange of material will take place from cytoplasm to the nucleoplasm and from nucleoplasm to the cytoplasm. That is a nuclear. Then last comes chromatin. During the interphase stage, the chromatin network will be there. And this chromatin network are of a two type. Chromatin network is nothing but the DNA itself, a long chain of a polynucleotide. That is a chromatin. And are of a two type, euchromatin and heterochromatin. Euchromatin is lightly stained and transcriptionally active. Whereas the heterochromatin is darkly stained and transcriptionally inactive. That is the structure of nucleus. In the next clip, we will go for study the structure of chromosome. This chromatin network will cut down and coils and forms into a body called as a chromosome. And that we will go for study it into the next clip. I hope you have been understanding it.